Creatine has escaped the gym. It now appears in posts promising sharper memory, faster thinking, and a more energetic brain. The biological idea is plausible. Creatine helps buffer cellular energy, and the brain has expensive moments. But plausibility is not the same as a reliable cognitive upgrade. Two public meta-analyses tell a nuanced story. Memory shows a small positive signal overall, especially in older adults. Meanwhile, overall cognition and executive function do not clearly improve. If someone sells this as a guaranteed nootropic for every healthy young adult, the evidence is already being stretched. The first review focused specifically on memory in healthy people. 10 randomized controlled trials met the criteria, and 8 contributed to the pooled estimate. Across those trials, creatine improved memory measures by a standardized mean difference of 0.29 compared with placebo. That is a small average effect, not a dramatic transformation. The studies tested different forms of recall and working memory, used doses ranging from roughly 2 to 20 grams per day, and lasted from days to months. Combining them increases statistical power, but it also blends very different experiments. Age changed the picture. In the younger subgroup, covering ages 11 to 31, the pooled memory effect was 0.03, essentially no difference from placebo. In older adults ages 66 to 76, the estimate was 0.88, much larger. That contrast is visually striking, but it is not proof that age alone determines response. The older subgroup had high heterogeneity, meaning results differed considerably across studies. Smaller studies, different baseline diets, and different tests can make a subgroup look cleaner or stronger than it really is. A broader 2024 meta-analysis included 16 randomized trials and 492 participants, mixing healthy adults with some clinical populations. It found significant pooled effects for memory and certain timed attention or processing outcomes. Yet it did not find significant improvements in overall cognitive function or executive function, and several domains were rated low certainty. That distinction matters. A supplement can affect one test category without making the whole mind work better. Faster completion on a task is also not automatically better judgment, learning, or everyday functioning. Why might older or stressed brains respond differently? One hypothesis is that an energy buffer matters more when demand is high or baseline stores are lower. But the trials did not consistently measure brain creatine before treatment, and dietary intake was often missing. Vegetarians obtain less creatine directly from food, yet subgroup evidence is still limited. The honest mechanism slide, therefore, has two columns, a plausible energy system on the left and unanswered questions on the right. The mechanism supports further research. It cannot tell you in advance whether your memory score will change. If you are considering creatine, separate the established exercise use from the less certain cognition claim. Discuss supplements with a qualified clinician when you have kidney disease, take interacting medication, are pregnant, or have another reason intake may need individual review. Do not use a high-loading dose because a headline mentioned the brain. The memory meta-analysis did not find that higher doses produced a clearly better memory result. Most importantly, judge any personal experiment against something observable, consistent training performance, or a defined task, not a vague feeling that you should be smarter. The takeaway is simple enough to remember. Creatine may modestly improve memory on average, but the result is uneven and appears much stronger in older adults than in younger participants. Broader cognitive performance does not rise uniformly, and executive function remains unproven. The public trials are interesting, not definitive. Treat creatine as a supplement with a specific evidence profile, not as brain fuel in a marketing sense. If your real goal is memory, the strongest question is not, does creatine work? It is, for whom, on which test, under what conditions, and with what uncertainty. Baseline diet, age, sleep, stress, and the exact memory test may all influence response. Until trials measure those factors more consistently, a group average should remain a group average, not a prediction about one person's exam, job performance, or risk of cognitive decline.